Please remember to ask your questions in our Q&A, because otherwise we'll miss them out in our chat box. And now we'll get started with the next presentation. Rosa, are you there?
Aquí estoy.
Hola, Rosa. Bien.
Hi, good morning. Okay, well, let's start with the following talk. And Rosa Alicia Jiménez was a professor of birds about the hybridization, which explained the geographic range and distribution of this species. She talks about the hybridization and how it explains the discordance in the phenotype-genotype discordance in the species Atlapetes albinucha. Hi, good morning, and good morning to you all, depending on where you are. Welcome to you all. Thank you very much to the organizing committee of this symposium and to the Society for the Study of Evolution for creating this event where we can exchange and get to know the work that we do. We'll tell you about my PhD work, but first of all, I would like to tell you what motivated me to apply as a speaker. The biodiversity of Northern Central America is very rich, thanks to its geographical position, which connects the North and South of America, and its geological and topographical complexity that promotes the in situ diversification. However, it is an understudied region, and I believe in sharing how incredible the biodiversity in this region is. And my idea is to motivate the future generations through the study of evolution with examples, local examples, which allow us to relate what we learn in class with what we see in our surroundings. In other words, to present evolution as real and tangible, and not as something that we can only read about in books. I came to the study of evolutionary biology through my fascination with birds. When I started learning about birds and studying the bird guides to identify them in the field, when I was a student, one of the things that really took my attention was finding these pairs of species or subspecies which were very, very close to each other, but had some minor differences, such as this exercise of, find the differences. Fortunately, I've been able to study one of these species, which is the Atlapetes albinucha bird. In order to study these processes, I take into account different factors. One of them is the phenotype, which is the visible characteristics of organisms. Also the genotype, which is the genetic information that an organism has. And finally, the distribution, geographic distribution. There are cases in which the phenotype and genotype are concordant. And in that case, we really talk about concordance, actually, between these examples, such as the example of the Cardelina birds. These are found in Mexico and Guatemala, and there's a concordance in the two species. We see here as 9 and number 10. And there is a geographical barrier which separates them, which is the isthmus of Tehuantepec, and these are lowlands and normally separate this mountain species which are on both sides. However, the phenotype and genotype are not always concordant. Here with the example of these birds of this genus. So we see that there is a concordance in the phenotype and genotype, but on the other side of the isthmus in Mexico, the Violiceps and Vidifrons from the Ramosomia group have a discordance. So analyzing different genetic samples and analyzing different geographic analysis, we have found that one of the causes for this discordance is the hybridization. So it's the flow between the two species. Another example that we can find in terms of the phenotype-genotype discordance is the Atlapetes albinucha, which is this species has two main phenotypes. A gray chest, gray belly example, and a yellow belly example. The gray goes down to Ecuador, but the yellow one only goes to Mexico. So the gray phenotype goes down from Mexico all the way down to Ecuador. However, previous studies based in mitochondrial ADN have found that the phenotype and genotype do not coincide. There's no concordance. So there is no separate group, which is the gray one, but they're actually mixed. So researchers have proposed that this is due to an incomplete lineage sorting.
Ancestrales detenidos. Esta propuesta se ha hecho en base
This proposal has been made based on the fact that the information that was had is that these two genotypes are not found in the geography, given the Chiapas Depression or the Grijalva River Valley, which separates the two mountain ranges. However, there's another area of possible contact, which is here between Mexico and Guatemala. And it is this area which we visited in 2009 in order to carry out some sample takings.
en colaboración entre
These
la
trips were done in collaboration between the University of San Carlos of Guatemala and UC Berkeley when I was still student, a bachelor student here in Guatemala. I had the opportunity of participating. We collected 2 specimens of albinucha, and one of them is at the University of California, and the other one's here in our collection in Guatemala. With this, I wanted to mention the importance of collaborations, equal and just collaborations, fair collaborations, where both universities or institutions can benefit from the joint efforts, both in terms of the resources such as the sample collections, as well as the academic exchange, of which I am an example. I am a product because I managed to work with this collaboration with Berkeley, which allowed me to study and do my PhD there. So with these samples, with the specimens that were collected in 2019, it was only when many years later, when I was working as an assistant in bird collection, I found this, which not only has a yellow neck, but it also has some yellow in what should have been more gray. So I thought, hey, this is an intermediary between the two phenotypes, the yellow and the gray. So I ran and started showing my advisor, and he encouraged me to continue investigating, researching.
amarillo que el otro, ¿verdad?
The specimen that we have here in Guatemala is a bit more yellow than the other one, but it's not as much as the one that we found in Chiapas. So taking into account this evidence of possible hybrids, we've asked ourselves whether hybridization explains the discordance, the phenotype-genotype discordance. And in order to answer this, we studied the genetic structures and the barriers. We looked at the color base in order to understand the two phenotypes, and we analyzed the geography and ecology of hybridization of these species. So in order to do this, we collected several samples, studied the ones that we had collected in Mexico and Guatemala, but we also looked at the ones that other museums lent us, which contributed greatly to the study. So we managed to have 73 samples, 73 individuals, taking into account the whole geographic range and the two phenotypes. From these individuals, we sequenced mitochondrial DNA, especially the ND2 base region, and we saw the haplotypes associated with the geographies. So we have a group of haplotypes that are the west of the region. One is north of Central America, one Costa Rica and Panama, and the other one goes to Colombia. However, these haplotypes are not associated to the phenotype, and this is coincides with what we had found previously in terms of the mitochondria DNA. So these is haplotypes that come from the yellow individuals, and the ones with green are the hybrid. These results coincide with what we found at nuclear level. In this case, and analyzing many different variables, we found that the genetic groups, phylogenetic groups, correspond to the geography, given there being a first divergence in the isthmus of Tehuantepec and another one in the Motagua River Valley in Guatemala. And again, the phenotype does not coincide with the genotype. The two main groups of yellow phenotype are closer related, of course, amongst themselves. And then we have the hybrid genotype, and this is another analysis, analysis of principal components, where we look at the intermediary groups here. This is what we see with a green color. And in this analysis, we again find the influence of the Motagua River Valley in separating the two groups in principal component 1 and principal component 2, and then Tehuantepec isthmus in component 2. Then we looked at the genetic basis of the color, because we had proved that these individuals were hybrids. We did this with the exon capturing technique. We have more than over 1.5 million of variable sites. So the hybrids are intermediaries between the two populations. And then we chose these samples of the adjacent populations to be able to find sites that are associated to this change of color. And we've obtained comparative data of the differences with different metrics. It was very surprising to find only one SNP which had FST equal to 1, which means it's completely different between the two groups. And when we examined the data of the nucleotides, we found that they coincide. The ones that are yellow in Chiapas have one cytosine, the ones that are gray, one thymine, and then the hybrid, which is the same as Chiapas. And then this is the one for Chiapas. So this nucleotide is found in a gene which has to do with an UDP enzyme, an enzyme involved in lutein cycle, which gives yellow coloring in many species, including birds. The one at the top is involved in the degradation of lutein. Finally, we studied the ecology and of this hybridization and geography of this hybridization. So we saw the data of the hybrids with this gene that we found it's associated to color, and we also found that this is the mitochondria, which is the red line. So there is a more flow of mitochondria from the gray to the yellow populations. We examined the climate variables in the hybrid zone. We also found that the annual precipitation and elevation are the variables that have a bigger effect in this hybridization area, where annual precipitation is greater in the areas where we have the parental regions, populations, sorry. And we also found that the hybridization zones have different environments compared to the areas where we have the parental populations. So taking into account whether this could be a barrier, is what some researchers have found is that there has been different species of beetles in each one of these areas, and some species of vertebrates. like such as mice and reptiles, and some species of birds which we have studied. In order to finish, I would like to talk about this question: how are hybrid zones in neotropical mountains? With this importance of the local examples in order to understand about evolution. So when I think about hybridization zones, the first thing that comes to my mind is what I was studying when I learned about this, of course. And these areas of hybridization areas in the Great Plains of North America, which are very, very vast plain regions. And what I've shared with you today, what is in this box, is 425 kilometers, which is very different to what could have compared to what could be observed in other areas. And it would be interesting to analyze what these hybridization zones could be behaving in mountains and other areas. So does hybridization explain phenotype-genotype discordance? Yes. The divergence in allopatry associated mainly with the isthmus of Tehuantepec and the Motagua Valley. These two individuals with the intermediary yellow-gray phenotype are genetically hybrid. The enzyme UGT is a candidate to explain the difference between the 10 of 5 phenotypes, and this hybrid zone occurs in a narrow area with a higher precipitation and a lower elevation than the environments of the parental populations. Thank you very much to you all. Thank you to my collaborators, especially Dr. Rory Bowie. Thank you very much, Rosa. It was fascinating listening to you. Let's go to the question. There is one from Nancy Savera. The question is, in the first global analysis of SNPs, did you separate the neutral from the outliers? In which case, we could see in a differentiated way the signal which would come from the dynamic population, which provides from any pressure, selection pressure. In the first analysis, we analyzed all the SNPs. We did not separate them. Thank you. And there was a hand that somebody raised the hand. I think the hand is still there. Yeah. Okay, let's go to the next question in the Q&A box. Luis Rodrigo Alce Valdez says, thank you very much, Craig. Congratulations on your talk. The question is, in order to search for the genetic basis of color, is your article already published, or can we access your PhD thesis? I asked that because in my lab we're interested in doing something similar, so I don't know if you could share your publications in the chat. Thank you, Luis. We are about to send this to a magazine, but if you write to me, I'm very happy to share this with you. Tesania Vega says or asks, have you analyzed non-visual signals such as UV pattern or birdsong, which correspond to genetic data? Birdsong has been analyzed previously, and we haven't found any differences between the two phenotypes. About the UV pattern, we haven't seen that yet. And Mariana says, why, according to the mitochondrial markers, is the genetic flow asymmetric, more common towards gray than to yellow? The flow already also exists in the nuclear markers. I didn't have time to cover that, but thanks for asking. But the nuclear flows exist in both directions. In the mitochondrial flow, since we know that it is inherited from the mother, and it only shows half of the story. It only goes from the grays to the yellow. Well, that could be, but this is just one idea. It could be that this is related to the environment and how those which are gray phenotype inhabit in bigger ecological environments. So there could be a relation to that, but we need to explore that further. Thank you. Okay, we don't have any more questions. So thank you very much. If you have any more questions, we can write them. We will have 5-minute pause and we'll come back in 5 minutes. Thank you very much.